During fertilization, the sperm and egg fuse to form a diploid cell called a zygote. The zygote cells divide, forming a new multicellular cluster called a blastocyst, which travels to the uterus where it implants itself on the surface of the uterine wall. A few hours after the sperm and egg have fused into the zygote, it undergoes a process called cleavage, where it divides into a pair of two new cells called blastomeres. Those blastomeres keep splitting, so it turns into a loose clump of 4 cells, then 8 cells, and finally into a structured mulberry-shaped 16-cell cluster called a morula, with inner and outer cell masses. The morula cells are held in place by the zona pellucida. That morula gradually develops an outer cell mass of trophoblast cells and an interior cell cluster with a fluid-filled cavity at the core called a blastocele. As soon as that blastocele forms, the morula is no more, and now it's a water balloon-shaped structure called a blastocyst. Now, the cells making up the inner cell wall of the blastocyst are collectively called the embryoblast, because they'll go on to eventually become the fetus. The embryoblast cells cluster together at one end of the blastocyst in an area called the embryonic pole. Meanwhile, the trophoblast cells flatten out, forming a barrier called the epithelial wall. Fully formed, the blastocyst hatches from the zona pellucida around the end of day 4, and is ready to attach itself to the wall of the uterus. Trophoblast cells help it implant into the wall of the uterus by putting L-selectin molecules on their surface, which are proteins bound to carbohydrates that bind like a lock and key to carbohydrate receptors on the uterine wall. Once there, the trophoblast proliferates into the wall, and are called syncytio trophoblasts. Also, during implantation, the mucosal tissues of the uterus are in a secretory phase, which is where the corpus luteum releases large quantities of progesterone, which is a sex hormone that floods the body and reaches the glands and arteries of the uterus. The corpus luteum is located in the ovary, and develops in the same spot where the unfertilized egg was initially released. Responding to the flood of progesterone, the uterine arteries become large and coiled, engorging the endometrial layer with blood, which are the tissues that line the uterus. Alright, as a quick recap. Just a few hours after its formation at around 1 to 2 days, the zygote undergoes a series of mitotic divisions, taking us through days 2 to 4 and developing into a blastocyst around day 5. This new cluster of cells attaches to the outer wall of the endometrium, where it starts to implant and reach into the endometrial wall. Meanwhile, the endometrial tissues react to a surge in progesterone released from the corpus luteum. They become engorged with blood and fluids, swelling up in preparation for the blastocyst's arrival, and this takes us up to about day 7.